Hello, a warm welcome from IGT University. I am Dr. Ashok Yadav, Faculty of Agriculture Sciences. Today we are learning about the breeding of pineapple. Pineapple is believed to be originated in Brazil. The wild Brazilian pineapple, Ananas microstatus, is considered an ancestor of the cultivated pineapple. It is reached India during 1548. The generic name Ananas is derived from the Indian nana. In India, much attention has now been given in the development of field gene banks. The pineapple Ananas communis variety is an herbaceous monocotyledonous perennial tropical plant of the Bromeliaceae family. It is cultivated in more than 60 countries where its fruit presents great demand and economical importance. In Brazil, it is the fifth more cultivated tropical fruit. It presents a great option for the cultivation in the non-traditional areas such as the semi-arid region. Pineapple plants require careful management and present some morphological and physiological aspects which knowledge facilitates the understanding of its flowering mechanism. Among those aspects is the apical meristem that differentiates into the leaves during the vegetative phase but later undergoes transformation, originating the inflorescence and thereafter retakes its vegetative activity, producing the crown of the fruits. Flowering is very important for the plants since flowering is the first step of the sexual reproduction resulting in the fruit reproduction, which is the main objective for the economical explorations of the plants. Thus, several studies aiming to understand how that transition is controlled have been accomplished in the last decades in the several species, generating a great amount of information. The floral differentiation of the pineapple plants has another similarity. It can be triggered artificially by the chemical substances that are also related to the natural flowering. The flowering process of the pineapple plants may be better understood if the life cycle is known. The cycle takes from 12 to 13 months until the first fruitescence is produced, depending on the environmental condition and crop management. This cycle can be divided into three phases. Vegetative phase, number first, involves the period from the planting to the floral differentiation. Number two, reproductive phase or flowering and fruiting phase goes from the floral differentiation to the fruit maturation. Number three is the productive phase, begins at the productive phase but the continues after the fruit is harvested, involving slips and suckers development until their harvest for the planting. Among those phases, the less flexible is the reproductive phase. Regardless, flowering is naturally or artificially induced. Several factors influences the crop cycle such as climatic conditions, mineral nutrition, type and weight of the planting material and planting time. The susceptibility of the pineapple plant to natural or artificial flowering is related to the age or size of the plants. The pineapple crop is commercially explored due to the possibility of the controlling the flowering, favoring of season fruit harvests when the fruit trees are higher or the production throughout the year. Pollination in the wild pineapples are pollinated primarily by the hummingbirds. Certain wild pineapples are forageed and pollinated at the night by bats. Under cultivation, because seed development diminishes fruit quality, pollination is performed by the hand and seeds are retained only for the breeding. In Hawaii, where pineapple were cultivated and canned industrially throughout the 20th century, importance of hummingbirds was prohibited. The flowers of the pineapple are perfect with floral bracts, three short fleshy sepals, three petals, six stamens approximately half of the length of the petals, and an inferior ovary with the three carpels. The pineapple is a herbaceous perennial which grows to 1 to 1.5 meter tall, although sometimes it can be taller. In appearance, the plant has a short, stocky stem with touch waxy leaves. When creating its fruit, it usually produces up to 200 flowers, although some large fruited cultivars can exceed this. Once it flowers, the individual fruits of the flower join together to create a multiple fruit. After the fruit is produced, side shoots called suckers by commercially growers are produced in the leaf axils of the main stem. These may be removed from the propagation or left to produce additional fruit on the original plants. Commercially, suckers that appear around the base are cultivated. It has 30 or more long, narrow, fleshy, throat-shaped leaves with sharp spines along with the margin that are 30 to 100 cm long, surrounding a thick stem. In the first year of the growth, the axis lengthens and thickens, bearing numerous leaves in the clone spirals. After 12 to 20 months, the stem grows into the spike-like inflorescence up to the 15 cm long with over 100 spirally arranged tuberous flowers, each subtended by a bract. The ovaries develop into the berries which coalesce into the large, compact, multiple fruit. The fruit of the pineapple is usually arranged into two interlocking helices. Typically, there are 8 to one direction and 13 in the other, each being a Fibonacci number. Inheritance pattern according to the Collins 1966 or 1960 spineless from in which spines are restricted to the top few inches of the leaf and dominated to the spiny wild types. Further, many wild cultivars process spiny leaves. Ananas anonososis is also a source of disease resistance. The cultivar Pernambuco is donor of the good flavor and aroma. Tender, non-fibrous, juicy fruits, early fruiting, resistant to hurt and root rot. Queen cane be for the crisp, non-fibrous, deep yellow flesh, fruit early ripening. Red spinach is a good source of vigor, resistant to the wilt, hurt or root rot. Singapore spinach can be good donor for square, shouldered fruits or golden yellow flesh, and wild species are also a good source of vigor, resistant to the various diseases and pests. Spiny tips and spiny characters are the phenotypic expression of the single or pair of the alleles, with spiny tip being dominant. Homozygous 55 and heterozygous 5 and produce spiny tips, recessive and may give rise to spiny plant progeny. The piping and non-piping characters are controlled by the another non-linked pair of the alleles with gene P, that means piping being epistatic to the 5 and S. The homozygous PP genotype produces pronounced piping than the heterozygous genotype. Frequent the mutation of the 5 and S occurs. Ananas eridifolius is having SE genes of the smooth tips leaves. Pineapple breeding programs were now more focused on the fruit quality improvement for the fresh market. A superior clone should have high yield and optimum sugar acid ratio, high ascorbic acid, disease and insect resistant to the tolerance of the physiological disorders before and after harvest. Production practices are continually evolving to the meet changes in the resources available to grow the crops and adapted to the new low acid hybrids with their increased susceptibility to the natural flowering and greater need for the attention to the fresh food markets. The grower goal is to economically produce a consistent supply of the high quality fruits. But clone selection is not a creative method. Old smooth cane clones in French Guiana are not very different from the most modern clones. Most mutations are deleterious and non-significant positive nutrients have appeared. with the possible exception of the build strain selected by our Mexican colleagues. Many cultivated clones of the Hispanola Raza and Singapore Spanish have reverted to the spiny condition, and even such important variety as Singapore Spanish, Pergola or Perolera also well adapted to the condition of important local markets or zones of the production suffers from the strong defects as a proliferation of crowns and slips. In vitro techniques, its importance is probably more related to the multiplication rate per seed than to the particular physiology of the plants and the growth chambers. However, there remain some risk that the progress in homogeneity, quality, and quality obtained through the selection of specific subclones may be partially lost in the micropropagation process.
So today we covered breeding of pineapple. Next time we meet you, we will be exploring about the breeding of guava. Keep learning, keep growing. See you next time. Thank you.